Before I start, I choose level 3. This is the context of this experiment. This is the materials what we use in this experiment. This is the prediction of this experiment. First, increase the heat block temperature to 95 Celsius. Set a micropipette to 30 microliter and attach a tip. Draw up the wild type cell culture. Close the wild type cell culture. Dispense the wild type culture into the wild type cell tube. Repeat the procedure to transfer 30 microliter of the pancreas cell culture. Repeat the procedure to transfer 30 microliter of the added for cell culture. Repeat the procedure to transfer 30 microliter of the added 7 cell culture. Firstly, in the collect cell, we use micropipette because micropipette commonly used to transfer volume liquid in the microliter range. Draw up 10 microliter of 4x SDS sample buffer. Close the 4x SDS sample buffer. Dispense the 4x SDS sample buffer into the wild-type cell tube. Repeat the procedure to dispense 10 microliter of 4x SDS sample buffer into the pantry cell tube. Repeat the procedure to dispense 10 microliter of 4x SDS sample buffer into the Edit for tube. Repeat the procedure to dispense 10 microliter of 4x SDS sample buffer into the Edit 7 tube. The protein ladder already contains SDS sample buffer. Following, in the second of X sample buffer, we use 4x SDS sample buffer because it's for ideal preparing and loading protein sample into gel for polyacrylate gel electropolysis analysis. Next, the purpose of protein ladder is a separate the protein primarily by mass because the ionic detergent SDS denatures and binds to protein. Move all of your 4 sample tube to the heat block. Set the heat block timer to 10 minutes and press start. Now remove all of your sample tube from the heat block. Move all of your sample tube to the microcentrifuge. Briefly spin the tubes. Remove all the tubes. Moreover, microcentrifuge is an important piece of equipment of any lab. It used for spinning a variety of samples at high speed. Place the polyacrylate gel in the electrophoresis box. Fill the gel cassette with 1x running buffer. Now remove the comb. Fill the outer gel electrophoresis tank with 1x running buffer. Set a micropipette to 20 microliter and attach a tip. Draw up the protein ladder. Move the micropipette to the first lane of the protein gel. Dispense 20 microliter of the positive control into the second well of the gel. Dispense 20 microliter of the negative control into the third well of the gel. Dispense 20 microliter of the Edit 4 cells into the fourth well gel. Dispense 20 microliter of the Edit 7 cells into the fifth well of the gel. Other than that, polyacrylate gel electrophoresis is a powerful tool used to analyze RNA samples. Polyacrylate gel helps to examine smaller molecules better since small molecules can enter the pores. Connect the red and black cable to the power supply and the lid of the gel electrophoresis box. Turn on the power supply. Set the voltage to 150. Set a timer to 60 minutes. Start the power supply. Turn off the power switch. Unplug the electrodes. Inspect your gel. Finally, electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. Electrophoresis is useful for we can see how many different DNA fragments are present in a sample and how large they are relative to one another. This is my actual result after electrophoresis. Next, compare the predicted control with the correct controls. Positive control and negative control, two types of tests that give completely opposite response in an experiment. The main difference between positive and negative control is that positive control gives a response to the experiment, whereas negative control does not give any response. Lastly, Kumasi stain gel is commonly used to stain protein in SDS page gel. The gel are soaked in dye and excess stain is then eluted with a solvent. The reason we use sodium doclosulfate SDS in this assay is answer is B. SDS SDS is break down cell membrane and coats the entire unfold protein in a shell of negative charge. Question number 2. Why are samples heated before loading them on the SDS gel? Answer is C. Heating the sample speed up the protein denaturation process. This is important as protein need to be unfolded in order to successfully separate out according to size during the STS polyacrylamide gel electrophoresis gel. Question number 3. Why might you rather want to run the STS gel electrophoresis at a low voltage than a high voltage? Answer is C. A low voltage will generate less heat and provide better protein separate and band resolution. Question number 4. STS sample buffer contain glycerol which has higher density than running buffer. Why is why is glycerol included in SDS sample buffer? Answer is A. The higher density of glycerol create a denser sample buffer, ensuring that the protein sample sink to the bottom of the gel well. Question number 5. In this experiment, you aim to detect the presence or absence of a specific protein produced by the CCR5 gene. What protein were separate by size during the SDS phase gel electrophoresis? Answer is B. All cellular protein in the ladder and the pancreas and monocrophage cell line. And the last one is the feedback question. How confident do you feel about using the technique you learn? I feel very confident. Finally, this is the summary of protein gel experiment. It is important to summarize after completed an experiment. Gel electrophoresis is a technique used to separate DNA fragments according to their size. DNA samples are loaded into well at one end of a edges.